All right, this week is a continuation of some information that we did about NextPoint and their HHLA2 and its receptor antibody treatments for maybe a handful of different cancer types. But got some newish information that should be interesting. I want to do a really quick run through of background just to set the stage again quickly. So first thing, context. Immune checkpoint inhibitors. That is the basic class that these drugs fit into, and the sort of original and still very effective and impressive for a lot of cancers immune checkpoint therapy were PD-1 and PD-L1 inhibitors. So remember, the concept of an immune checkpoint is that normal healthy cells have to have a way to tell white blood cells that they are healthy and they should be left alone. And so in this particular case, the tumor cell would express PD-L1, right? L being a ligand for the receptor, PD-1, which you would find on either a T cell or a natural killer cell, which are the specific cell types in the immune system that typically go after unhealthy, deranged cancer cells. Okay, and so we've got this battle lined up between a T cell or some other white blood cell subtype that's recognizing a specific antigen on the surface of a tumor cell. This is a positive thing for us in that it encourages the T cell to attack. On the other hand, the tumor cell is trying to use PD-L1 to tell the white blood cell, no, leave me alone. I'm a perfectly healthy, normal cell. Go about your way. The more of this we have, that's a negative, meaning it inhibits the T cell from going after the cancer cell. And so we've seen a handful of different therapies that target the immune system for treating cancer, but almost all of them come down to one of these two pathways, right? We can either stimulate the T cell by, teaching it to recognize more antigens on the cancer cell. That's where like CAR T cell therapy comes in. Or we can try to inhibit this immune checkpoint. And that general strategy is called an immune checkpoint blockade. Right? We're trying to block the checkpoint from engaging so that the T cell is released to go and attack our tumor cell. Okay. The follow-up to that is it's more than just PD-1 and PD-L1. Those are the most common targets. We've already mentioned those. There are a handful of antibodies, some of which target PD-L1 on the tumor cell, some of which target PD-1 on the white blood cell, and at least one example of the CTLA4 antibody that blocks a different ligand receptor pair that's also considered part of the immune checkpoint system, just separate from PD-1 and PD-L1. All right, so our new receptor and ligand HHLA2 just add more to the story. Basically that there's redundancy built in. It's not entirely clear at this point exactly how PD-L1 versus HHLA2 really differ from each other, but they both have that same effect. It's just they're not all ubiquitously expressed. So that just means not every cancer cell expresses all of these molecules. Some are very strongly expressed in certain cell types, less so in others. Okay, so quick recap. HHLA2 is the equivalent of PD-L1, right? That is the ligand that is on the surface of the tumor cell, maybe. Again, not 100% of tumors and tumor cells within one tumor express it, but many do at fairly high levels. All right, KIR3DL3, horrible name, but that is the receptor that is expressed on either natural killer cells or T cells. Okay, so in order for the tumor to effectively escape the immune system, both of those components have to be true for this mechanism anyway. The tumor cell expresses HHLA2, and the T cells and natural killer cells that are infiltrating the tumor, right, that are actually found surrounding those cancer cells, have to have the receptor. And so it seems like a lot of cancer cells, especially the most resistant ones to these therapies and to the immune system, are expressing multiple immune checkpoint proteins, right? HHLA2, PD-1, possibly others. right, to try to activate as many of these receptors as they can on the white blood cell to initiate that immune checkpoint pathway and hopefully to them be left alone. So our goal is to block this interaction, right? And NextPoint has two different antibodies that I did a little bit more digging and found that they are both in early stage clinical trials. So there's NPX 267, which is an antibody that binds the KR3 receptor on white blood cells. And there's NPX 887, also a monoclonal antibody that binds HHLA2 on the actual tumor cell. Okay? So if this is going to be made use of, then we need to find patients that have higher levels of HHLA2, which is also called B7H7, same thing. And in order to do that, right, we need some sort of biomarker. And so this biomarker is key to the paper that we're going to look at in a little bit. It's also key to the clinical trials that these antibodies are being sent through because it's used to determine which patients are most likely to respond favorably to the treatment, particularly those that have the highest HHLA2 levels, because that's clearly part of their strategy for survival. All right. So I did a clinical trial search within the U.S. system anyway, and interestingly, there currently appears to be no competition. All right. So there are two different clinical trials currently out. There's the NPX 887, which binds HHLA2 correctly, and this clinical trial is still in phase 1. It just started in late January of this year. It seems that they're still recruiting patients, and the completion date isn't expected for 3 to 4 more years. So early stage, still going, but they're in the safety testing phase. There's no realistic expectation that we're going to get results back anytime soon, unless, if the results are absolutely spectacular, it could potentially be before that 2027 date. But with the time frame that we're talking of, hoping to get these patients to survive for years after their diagnosis and treatment, that seems unlikely. All right. So just as a reminder, phase 1, we are effectively at dose escalation studies, just to make sure that it's tolerated and that it's safe. So at this point, they're not even really looking for efficacy. All right, the second one, which started slightly earlier, but still second half of last year of 2023, this is targeting the receptor of HHLA2, right, which we already talked about. So different antibody, same general strategy, same pathway that we're blocking, also currently in phase 1. Projected completion date is about 2 years out. This one started a little bit earlier, and it could just be the patient population they have, that its timeline seems to be a little bit moved up. But same deal, still in dose escalation. They're not at the point of administering whatever they determine to be therapeutic doses quite yet. But I would assume that should transition probably by the end of this year, I would think. Right. So 2 clinical trials in process, and this poster we briefly saw before. I found that a lot of this is based on data that was published over a series of papers, but a lot of it came from a science paper that I managed to track down. So I'm not going to jump back into this exactly because I got more interesting information, and that particular paper was published in a science journal several years ago. 2021, I believe, was the publication date, and everything matches up. So a lot of authors on this list, and it seemed like it was a collaboration between a handful of different universities and hospitals, and interestingly, NextPoint is on the author list. So this guy, Zhang, who is his primary appointment seems to be at Albert Einstein College of Medicine, where he's a research professor, and a nearby medical center that's also in New York. And he's listed under those titles and at NextPoint, so it's pretty clear that he's still working in his clinical research role at the university and the hospital, and effectively founded NextPoint as a startup on the side. And it seems like this paper was the big splash that sort of pushed these potential therapies into the realm of possibility, but it very much seems like this guy's thing. There don't appear to be any other competitors that have antibodies in development, definitely nothing in trials, nothing that I found when searching. So it seems like if this works and it's effective, they have a complete run to something marketable as far as sort of cornering the market on IP goes. Okay, so big picture. Obviously, we want to find out if, one, tumor cells express high levels of HHLA2, which the answer is obviously yes, or we wouldn't be in trials. And then second, can blocking HHLA2 or its receptor decrease cancer growth? Obviously, we can't try this in humans, and as has become very, very popular, they're going to use effectively what's a xenograft model, where they take human cancer cells and implant them into mice, and then use our experimental antibodies to try to slow that growth. Right? And along the way, we would probably like to know how T cells, so CD8 T cells, are what we're interested in, because they are the ones that recognize antigens on tumor cells, and NK, natural killer cells as well. So this paper had a massive amount of data detailing, which white blood cell types the receptor is found on, what kinds of tumors and cancer types is the HHLA2 molecule found on, and then how effective is it at slowing different cancer types. Okay, so the first question, for a handful of different cancers, right, they happen to pick kidney, lung, gallbladder, and gastric, which is stomach cancer. And we're looking at a handful of different things. So this panel is measuring characteristics of white blood cells that were taken from those tumors. Because remember, if you extract a tumor from a patient, you are going to have white blood cells that go along for the ride, because they were infiltrating the tumor and trying, but clearly failing, to fight it off effectively. Okay, and so in these stained images, they used 3 different fluorescent colors. KIR3, DL3, remember, is our receptor that you find on white blood cells. This CD45 is a ubiquitous white blood cell marker, meaning all white blood cells have CD45 on their membrane. It's not really variable, and so it's a good way, when you're looking at a mixed population of cells under a microscope, is to use some kind of fluorescent tag like that, so you know what the total number of cells is that you're looking at. That can tell us what percentage are KIR3, DL3 positive. Okay? And DAPI is just a DNA stain that's staining the nucleus. It's used in pretty much every cell type, just a way to know where are all the cells in the population. So DAPI stains every cell in the tumor. CD45 specifically stains white blood cells. And of course, the KR3 specifically stains cells with the HHLA2 receptor. All right. So what we want to do is a merge, right? We do them separately with each channel, and then you overlay them. And what we're looking for is an overlay between green and red. It's a white blood cell, and it has the receptor that we're interested in. So this zoom in is showing you anywhere that yellow color shows up. That's the green-red overlay. Those are our positive cells. Cells that are red that don't have green, right, which I guess on here would be these guys that are bright red, those are white blood cells that lack the KR3 receptor. All right. At the same time, with a slightly different method, they also stained the tumor for HHLA2 and PDL1 mostly as a control because of its known role in immune checkpoint suppression. So the dark staining that you're seeing, the light blue are nuclei. The darker this brown staining is, this tan staining, the more of our protein target there is. And so you see relatively high levels of HHLA2 in effectively every cancer type that they studied, compared to relatively small amounts of PD-L1. And I don't know if they specifically chose tumors and samples that were PD-L1 negative or low in expression, but the HHLA2 quantity is clearly dramatically higher. Okay, so next question, how much expression is there? So this is, the y-axis is HHLA2 expression levels, and specifically for blue, right, KIRDL3, those are in cells that were CD45 positive, meaning white blood cells. So this is in tumor cells, right? Do they have the ligand? This is in white blood cells. Do they have the receptor? And then again, PD-L1 as a control comparison just to see whether levels of HHLA2 are higher and lower. And you can see for many of these cancer types, significantly elevated HHLA2 levels over PD-L1, right? Except for, I would say, maybe the lung cancer is relatively equal between the two. One word of caution, just because a particular protein is expressed at higher levels than another doesn't necessarily mean it's more important. So all these data really show us is that these 4 cell types, cancer cell types, express high levels of PD-L1, and a certain subset of white blood cells in those tumors express the receptor, that KR3. So this gives us hope that there's going to be some effect in blocking the pathway, because we assume that tumor cells wouldn't upgrade a molecule that they're not going to use. And the high levels of HHLA2 suggest it's giving some advantage. Otherwise, right, why would it be there? Okay, so to figure out if it's effective, we're going to do a xenograft study. And that involves taking cell lines, human cancer cell lines, injecting them into these mouse models of cancer, and they're specifically injecting cells that have a luciferase reporter gene, this LUC2. That luciferase produces light when you add the substrate to the enzyme. So basically, these tumor cells glow in the dark. So we can inject them into the mouse. You can put the mouse in this whole body scanner that emits, or that detects light being emitted by the bioluminescent tumor that we injected into the mouse. And the heat map over here is showing you intensity. So blue is relatively low amounts of luciferase, green somewhere in the middle, and then yellow-red on the heat map, highest levels. And so you can see pretty much every mouse started off in a fairly similar position when they were first injected. And by the end of the study, the antibodies, right, they're testing different antibodies that block HHLA2 or its receptor. You can see that they showed significant reductions in tumor volume in a lot of cases, especially for this 26E10. All right, so these data, right, with the luciferase quantified over time, are what are producing these graphs. Okay, so total flux in this case is representing total amount of signal, like the amount of light coming out of the mouse, which we assume is directly proportional to the number of tumor cells present, right? And in both cases, our antibody showed significant improvements, right, over the control antibody, suggesting that for some of these cells, at least a subpopulation, at least, they significantly suppress tumor growth. Right? Now, unfortunately, the tumor did keep growing, but it took a hit. And so, like, based on those results, my guess is I don't imagine that this antibody is going to be used in isolation. It seems like the perfect pairing for an anti-PD-1 or anti-PD-L1 antibody, right? Trying to hit those tumor cells and the white blood cells that are going after them from multiple angles to try to reduce the immune blockade in the tumor, so that those T cells and natural killer cells have every aspect of the checkpoint released so they can attack the tumor. Okay, last thing I wanted to include, just because, I stumbled across it. I'm not sure if you saw this. All of these trials are definitely not going to be cheap. And they had this this article posted in a link on the NextPoint website. So they're $80 million in funding, which is obviously a sizable amount of money, but for a cancer trial, it's nothing insane. That money, interestingly, came from a program funded by Bayer Pharmaceuticals that seems to be used to try to acquire a decent stake in new companies that they think have strong potential. So anyway, the founders of this company very clearly came from the academic research side, but it seems like at this point, their backers and their money are definitely coming from the bigger side of pharma. So hopefully we'll get results back before too long, and we can take a look at data from those trials at some point in the next several years.